Hi, my name is Carmela Bangkong. For the 2021 Undergrad Symposium, I will be talking about isolation, identification, and characterization of biofilm microbial communities in 5 plastic polymers incubated in distinct freshwater ecosystem. Starting off with a little bit of background, plastic in the modern world is a necessity. Its ability of being lightweight, inexpensive, durable, used in a multitude of applications within the medical and technology industries have made it a formidable polymer. To put plastic production in perspective, as of 2017, 8,300 million tons of overall plastic was produced. Approximately 1.15 to 1.24 million tons of plastic entered the ocean from rivers annually, and about 10,000 tons of plastic enter the Great Lakes every year. This is becoming an issue as current waste management pathways only have 3 main modes to dispose of plastic: recycled or reprocessed, thermally destroyed, or disposed of in landfills or the environment. Again, these methods are not 100% effective or efficient. Recycling is often mismanaged in a lot of countries. Secondary materials are usually lower in monetary values, but require high and more expensive technical work. Thermally destroyed plastic have adverse health and environmental impacts. Lastly, not only is landfill upkeep expensive, discarded trash has been found to have detrimental effects on the environment, of which 60% of discarded trash ends up in the environment and only 10% really get recycled. Accumulating microplastic debris in freshwater environments is gaining recognition worldwide. Especially the pervasive nature of microplastic raises concern of its effect on the environment, terrestrial, and marine organisms. For this research, we focus on the Laurentian Great Lakes, which accounts for 1/5 of global freshwater supply and about 84% of North America's surface freshwater. Freshwater ecosystem is specifically important as it pertains to the global freshwater cycle, water supply, and human health. Plastic debris, therefore, is highly relevant as its composition and structure favor biofilm formation. Different polymers may select for different microbial communities, and there's even growing evidence suggesting plastic-associated microbes may harbor pathogenic, toxic, and remediative bacteria. The co-occurrence of wastewater contamination and plastic debris may even promote the formation of plastic and antibiotic-resistant biofilms. Because plastic debris is made up of various polymers and additives that enter the environment through a variety of pathways, the variability in biofilm characteristics, especially pertaining to pathogenicity, antibiotic resistance, and ecosystem function, is still largely unknown. For this project, our objective was to evaluate antibiotic resistance of culturable bacterial microbiomes associated with plastic polymers aged in different freshwater environments. Alongside the hypotheses, bacteria presence and pathogenicity is dependent on the smoothness of polymer surfaces and environmental conditions, where greater surface roughness promotes refuge for pathogenic bacteria and more contaminated systems have more pathogenic species. In this research, we use traditional molecular biology techniques. This is a quick flow diagram of the method used in order to tell us whether plastic type and/or environment influences impact the potential for harboring and/or be a potential vector for antibiotic-resistant bacteria. 5 post-consumer polymers: polystyrene, polyethylene tab phthalate in the form of bottle and fabric, and PPE in the form of polypropylene disposable masks and nitrile gloves, were incubated at 2 water bodies in western New York for 4 months. We used a stormwater retention pond on the RIT campus and an abandoned quarry pond at RIT's research preserve. Both systems are directly connected to Lake Ontario. Polymers were incubated and set up in a floating and weighted down rigged setup on the surface and benthic levels. 0.5 square inches of plastic swatches incubated from our study sites was inoculated in minimal media Reasoners 2 agar and rich media tryptic soy broth for 24 to 48 hours on 30 degrees Celsius at 100 RPM. This was followed by a series of tenfold serial dilution and restreaked for isolated single colonies based on morphology. Polymerase chain reaction was conducted to amplify the V3-V4 regions of the 16S ribosomal RNA gene, which is a genetic indicator to identify bacteria at the genus and/or species level. This can be confirmed by the presence of band around 500 base pairs. Purified amplicons were sent to GeneWez to generate sequences. The basic local alignment search tool, also known as BLAST, was used to identify genus species of bacteria. From our sequence amplicon, the bacteria genus Pseudomonas species was the most common across all polymer types, and MASC had the highest number of unique genera. Bacteria in the Pseudomonas genus are responsible for a number of clinically relevant diseases such as urinary tract infections and respiratory system infections. Once we were able to identify present bacteria, it was subjected to an antibiotic assay using the Kirby-Bauer disk diffusion method in order to determine the presence of antibiotic-resistant bacteria associated with plastic polymers. We used 10 clinically relevant antibiotics with tetracycline as our positive control and water as our negative control, some of which resulted in total resistance and/or susceptibility, which looks like this. Resistance and/or susceptibility to antibiotics is measured in a zone of inhibition. The smaller the ZOI, the greater the degree of resistance to the antibiotic, and the larger the ZOI means greater sensitivity to the antibiotics. We found that most isolates in the study are resistant to antibiotic classes beta-lactams, synonymous to penicillin, and glycopeptides, lipoglycopeptides, synonymous to vancomycin. Isolates in the study showed resistance to at least one antibiotics used, suggesting that antibiotic resistance is common among bacteria on plastic biofilm, at least for the species that are easily cultured in a lab setting. From this preliminary study, our results indicate that plastic pollution in a freshwater environment has the potential to harbor bacteria that are resistant to clinically relevant antibiotics. PPE is a growing type of plastic debris in the environment that may serve as an additional environmental refuge for resistant bacteria. Isolate resistant to beta-lactam antibiotic class may have further implication for human health, the ability to treat bacterial infections, and local water supply. Additional work is needed to tease apart whether individual polymers types are more likely to harbor pathogenic and/or antibiotic-resistant organisms, and whether some water bodies are more likely to promote the growth of these species. Finally, I want to acknowledge New York Sea Grant College Program, National Oceanic Atmospheric Administration, Emerson Summer Undergraduate Research Fellowship, and my advisors Dr. Christy Tyler and Dr. Andre Hudson for the support and continued support of this project.